Is
Yeah,
that possible for us to finish it off by 7:30 in an hour? Is that possible?
yeah, I will try to finish it within
Yeah.
that because
Please
to
try. Yeah, it's already 20 minutes late, so we
yeah.
request you to try to complete it by 7:30 Indian time.
Okay, okay. Also, please like remind me at 7 because
Yeah, sure.
sometimes while I was at
Definitely. I
teaching,
will do.
I'm not able to see the time.
Okay.
So yeah, okay. So good evening, everyone. Let's start with what type of data we are going to deal with, and in bioinformatics analysis, gene expression analysis, what type of data we are going to work with, and then databases from where we are going to retrieve these data. So from where we are getting this data, first we need to understand, and for that we have the central dogma of biology here, where from the cell, within the cell we have nucleus, and then there are chromosomes compiled with the like long DNA sequence, and on that DNA we have a small sequence where the genes are stored. So basically the small section of the DNA which is coded with the four basic nucleotides, ATGC nucleotides that we have, so that the combination of those nucleotides makes a gene. So that can be like length can be longer or shorter for different genes. So we get the gene from there, and for these genes we are trying to analyze the data, and gene expression we are getting from these genes only. Whenever like for different functions in our body, we have different function for specific genes for each function. For example, someone has black hair, so they have the gene on or off, or some other gene is working for that colors, and then eye colors for height and other thing. So genes are functioning, and based on their expression, how they are expressing the physical appearance will be different. Okay, so this is basically the origin of data, and how we are getting the gene expression data actually is whenever we are getting, if we are doing our DNA-seq analysis, so we will extract data directly from DNA, but when we are doing RNA-seq analysis, so we will extract the data from RNA, that is with the nucleus. And then from DNA, the samples, the patient or whichever sample we are getting, we will get from the concerned patient. And then in within the wet laboratory, the data preparation will be there. So DNA library will be there, adapters will be added to your data to stabilize your data, because the DNA in our body is in very active form, so it will not be able to like access the expression value that we are going to analyze. So for that, different library preparation will be there within the wet lab, and that prepared data or the semi-processed data will be going to the sequencers. So you all know like what type of sequencer we have. Illumina or Sanger sequencing process is there, and those type of different sequencer is there. Nanopore sequencing technology is there. So from each sequencing and technology, we have different type of data, and maybe some sequencers are able to give long reads. And some are able to read only short length RNA sequences or DNA sequences. So based on the sequencer also, sometimes our data depend that what type of data we are going to get from our sample, because from same sample, if you are using different sequencer, then you may get varied like data in your raw data file, which is in FASTQ format. Then there are different types of file format that we need to deal with when we are doing gene expression analysis, in which some of them are FASTQ, BAM file, VCF file. These are just the general example, but we have these type of files which have different informations in different formats. So this we will check further, but this is how the process actually works. So like the data is not coming from like any random place, it is just from the human body or the any organism data that you are going to analyze. It will be processed and go through sequencers to get the raw data that we are going to use from different open source libraries that we have, or the databases that we have. Okay, so first we need to understand the file formats. What are different file formats we have? After file formats, we will see that from where we can get these files actually. So first we have the sequence data. Like here in the second figure, you can see the nucleotide sequences are given in the second row after the header. So header will give the information about which organism you are using, or which on which machine or the sequencer it has been processed, so that whenever there is a difference in your FASTA file, so you can understand that okay, it is processed on this processor or this sequencer. Okay, and then sometimes the header have more information, like which chromosome, from which chromosome this particular sequence belongs. So header also varies based on the sequencer. Some sequencers give complete information in the header for what chromosome it is, and then what sequencer name is, what is the like front forward reads or the reverse reads. Like the sequence is read from the starting point, like 5 prime to 3 prime or 3 prime to 5 prime. So these keywords you might already be aware of. If you don't know about 5 prime and 3 prime, that also you can just drop your query in this discussion forum, or you can try to also explore, then I can answer there. So FASTA format, we have FASTA format with file extension .fa, .fasta, or .fsa. Okay, so all three formats will have the similar kind of data. Like first row will be the header and second row will be the sequence. So every time the sequence will be followed by the header, because header is having important information about your chromosome, about the sequencer on which this data is processed. And then in sequence line, you can see that ATGC nucleotides are given there, but the sequences are different for sequence 1 and sequence 2. You can see the sequence 1 is starting from different nucleotide, and the sequence 2 is starting from different nucleotide. So that depends on gene to gene. Like sequence 1 is for another gene, sequence 2 is for another gene. So this is how the FASTA format looks. When you have the FASTA file in .fa format, you can open it, or in text file format also, like with the notepad or any other text file, you can open it and you can visualize very easily this. And there are also some web-based tools where you can open your FASTA file in text format, or like visual presentation, you can open that. Okay, so if anyone is oh my God. So this was happening actually again and again. So let me just share you the PDF of my screen. Just please bear me with. I'm sharing you the PDF. Just a minute. Okay. I need to see if it is appearing in PDF also. Okay, we have it in PDF. So just give me a minute. I am sharing it with you. Share. Where is the PDF? Okay, I will. Okay, just tell me if you are able to see the PDF or not. I have opened it. Can you see the PDF? Please text down in the chat box so that I can understand. Oh, where is
Yes,
it?
Dr. Ghazala, we can see the PDF.
Okay, 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 that's great. Let me see. Like whoever has query, like any query in between, please drop down in text box so that I can understand that you are following with the lecture. Okay, so here is the, okay, so FASTA file we have already discussed. Now we will, okay. Now we will discuss about FASTQ file format, where again in FASTQ file format, we have the sequences in our data, but this file will have 4 rows. Okay, so first row is again the is called label, but it is again the header where the information will be saved for your sequence, like chromosome number and the sequencer number. So like here you can see in the bottom figure. Okay, do I have the pointer or something? I am not sure. Okay, so in the figure, in the this figure, we can see the header has the information for the instrument name, which is the sequencer, and then flow cell info that is on sequencer, like which program is activated. Then what is the cluster file name? So that is all information related to the sequencer, which is on which this sequence or the sample from the patient is processed. And then within that patient you can see like which the sequence like order for the nucleotides. So it starts with GTT like I'm showing the figure which is at the bottom. So we can see the nucleotide orders. Okay, and then third row will always be this plus sign or addition sign will always be this every FASTQ file. Okay, if this is not there or any FASTQ file is not following this format, it means there is some error in your FASTQ file, and that will not be like processed successfully on the files that you are on the tools that you are going to use for your data analysis. Okay, so always like we whenever there is some error or something, we try to open our FASTQ file and then see like is it the data which is stored there is following the order of the FASTQ format or not. If not, then we need to reconfirm from the database or the origin from where we have downloaded. Okay, or try to download the same data set from other database. So this is the format, and the fourth row is the quality of your sequence, like how much. So this quality score is are the ASCII codes you can see. So there is a ASCII table where for each symbol, and there will be sometimes there are numbers, like you can see at the end there is 3, then 1194, and there are greater than, less than, equal to symbols. So these type of symbols are stored in ASCII table. So that we need to you don't need to understand actually these scores, but you just need to understand that there will always be the fourth row in your FASTQ file with the scores given in that row. If fourth row is not there, it means it's not a FASTQ file or an incomplete FASTQ file. Okay, and these scores are the given based on the this Fred number. We call it as Fred quality scores, which is calculated as minus 10 log 10 of p, that is the probability of the error in like identifying this particular nucleotide on your first chromosome, or first, or what, whichever chromosome number is there or the gene number is there for which this specific sequence is. For that, the score will be calculated. So for example, if there is a gene nucleotide at the first position, you can see the figure at the bottom, the first nucleotide is G. So for G, what is the score for G? That it is, so if it is like what is the accuracy of like identifying this particular nucleotide in your data. If the score is more, it means it is like significantly shown or the identified in your data. Okay, so based on that, we need to see that just we are trying to understand these formats, because we need if there is an error, we should not tell or understand that tool is not working. Okay, there must be problem that your data is not correct. Okay, so you need to be sure that the data that you are using it is
Neighbor
good. And sorry. Okay, please mute yourself, like the participants, so that it should not create any kind of issue in between. So yeah, so why we are trying to understand, because no one opens the FASTQ file, like generally the beginners, like whenever we start learning or something, so we don't usually open FASTQ file. But why we are understanding, because this is important. If the error is there or you are not able to identify number of genes or good number of genes, or there is error, we should not say that the tool is not working on my data, because tool is made for this type of data. Okay, so there are tools that is specific to process FASTQ file. So why tool will not work? maybe your data is not correct, right? So we need to know that our FASTQ file must have 4 rows, and what each row will follow, and what is the pattern of FASTQ file, that we must need to understand. But it is not necessary that you should understand what are the ASCII characters given for your nucleotide and what is their value. And if it is like, never in your life, maybe while analysis you have to see that why 99 is there at this particular place, it should be 10 or it should be 8 or 7, because this is the task of the sequencer. Okay, so you are not going at the sequencer end. Sequencer is already programmed and made it to calculate this the scores with the program that it is given. Okay, so you just need to make sure that your data is correct, okay, the data that you are downloading. And yeah, so another thing I will say that whenever we are downloading the data, you are not supposed to open the FASTQ file that you are downloading and see each FASTQ file, because sometimes we are working with 10 samples, sometimes we are working with 80 samples. So 80 samples you will not download and open to see like your data is correct. Most of the time data is correct itself, that's why it is uploaded. But sometimes it happens that there is incomplete data, or sometimes while uploading, the whoever was uploaded, the owner was uploading, sometimes the error happened in between and then incomplete file was uploaded. Then only these type of issue causes that your data is not in correct format. So sometimes if your tool is not like giving error and error again, and some tools also give proper like clear uh, uh, instruction that maybe this error is because the header is missing in your FASTQ file. So you can clearly understand that your FASTQ file is incomplete or not properly given for as in the header line. So you can open the file and you can see at that time. Okay. Okay, next is VC file, which is called variant calling format file, the short form for this, and the file extension will always be the file name with following the .vcf. Okay, so this will be the VCF file, and its format is like on the first few top rows will be VCF header file. Again, in the header it will be given as the whatever the tool which is used to process this type of file, so that about that file and the date and the version of the tool, everything will be given. The organism will be given, that for which organism this data is. And then after few rows, after the header completes, will be the main body of main body, which is called as data, the data that you need to further identify the mutations or like SNPs or insertion deletions. So those are the type of insertion deletions, and SNPs are type of mutations in the genome. Okay, so if you are processing the mutation data, or you are trying to identify mutation in your data, like mutation, you must also know that the alteration in the nucleotide on the same gene, like on the gene, if there was originally, you can see in the figure, the first row in the body is ACG. If it is visible to you, that will be great, because here I have added that in the reference allele, REF stands for reference allele. So if it is reference nucleotide, it is ACG, then alternate, it has been changed, okay, due to some issue, maybe environmental issue or the hereditary causes, and the allele is changed to only A, or sometimes it is showing or captured by the tool as AT, but it is not ACG actually, so it is changed. This change may cause the disease, or we are not sure about disease, so for to like say that this change is exactly causing this disease or something, so for that we need to do further processing, like we need to annotate the data and all, but we can see clearly that the allele is changed. So some change need to be noticed, and we should further explored, so that we can see if it is like significant or not for the study, if we are studying for some disease. And then other columns we can see chromosome number is there, position ID is there, that on which chromosome this particular changes is happening. Okay, and then other information, that filter pass is given there. For that, if the this the tool that is used to identify that this alteration is happening at your chromosome number 1, so like this is passed that yes, it is like based on some scoring values, like out of 100 it is give, if it is showing 80 times, then it will give pass to everyone, every alteration that is shown here. Okay, so this type of different columns are there, given there, but most of the time we use here the first few columns, because there we can see clearly the difference in the reference and alternate alleles. Then again, for some scores are given for different samples, like samples are given sample 1, sample 2. So like we use healthy samples and disease samples, so for each samples different scores are given. If there is a reference allele with GT, then score is 0. If alternate alleles, then GT is greater than 0. Okay, so these scores, like most of the time you don't need to like be worry about. You just get the reference allele and alternate allele, get the chromosome number and the position, that on the chromosome which position like this alteration is happening. And then you can further process this data and see that in literature survey or in further database you can use, where or further other tools you can use to analyze that what this alteration is happening, that is this alteration also found in other disease, the disease which you are studying apart from that. Okay, then after processing few files, like fast few file if you are processing, then after get SAM file in between, like processing, you get SAM file and BAM files. Okay, so SAM file is like text format data, where you will have the tabular data, or text format data where you will have read name, then a position of the flags will be given, that okay, this is like based on the score, that what is the score of that particular like sequence that is identified, the read sequence that you have, for that the score will be given, position will be given, that on the chromosome 1 what position actually, like after how many nucleotides this particular read sequence is available in your data. Okay, so based on that these particular informations will be given, quality is given, for mapping quality is given, so this map q is mapping quality. So when we align like okay, so these terms you may not understand, but let me just tell you, when we will do analysis you may understand. So when we align our sample, for example you have the disease sample and you align it on reference genome, and reference genome is the universal healthy sample. Okay, sequence, healthy sample sequence. Okay, so on reference when you align your disease sample, then you will see some differences that I will show you in the next few slides, that how we see the difference when we align our data. So in that, there we use like this see the what is the quality of mapping, like how much it is matching to the reference genome, the data that we have. Okay, then further metadata is available, where metadata will tell you that what is the age of the patient, or maybe there is any like drug consumption history of the patient, they have been taking alcohol or something, or any type of additional data that is given for your sample, that is metadata. So these are the columns that are present in the SAM file. Okay, but SAM file, because it is a big file, we have a lot of number of genes, and then for each gene we have this number of information, so it is not easy for us to visually understand and differentiate between two sequences, right? How long we will read this sequence, GAA CAA, right? So for that we convert this format, SAM file format, in the BAM format, so that we can visually see that what is happening exactly in our data. Okay, so that we will see later. So you just need to understand, if anytime your tool is giving wrong answer, you need to open your file and see if these columns are given in your data or not. If anything is missing, you need to see like if the data or the column which is missing, is that significant for my data and it is missing, that's why the error is being thrown, or what is the actual issue? So for that only we need to understand these file sequences. Okay. Next is BAM format. So SAM file that we just saw in the text file format. Okay, so this file is converted to binary format. Okay, so this binary format we can now visualize. So, and this file will have the extension .bam, where it have the columns, queue name, flag name. So similar columns will be given. Additionally, binary information, because we are converting it into text data into binary format. So additional few columns will be there, because that is using to convert this text data into the binary format so that it will be visualized. Okay, so just it is the whole information that was stored in BAM format, SAM format. We are converting using the SAM tools into the BAM format. Okay. Okay, this was the bed file format again. This is happening. Okay, so bed file, maybe I will explain you later. This is bed file format is also similar to BAM format that we can visualize. It's just that few columns are extra in the bed format. So this is again the technical issue, but this is like you can understand that it is very similar to BAM format, and it is also binary format. Like bed format is also binary format, which we can visualize and see like what is the difference in our data. Okay, and the extension will be bed format. I will show you in the next session maybe, or I will check in the PPT if it is available now. And yeah, so here is the BAM format where we are using the IGV tool. Okay, so there is a tool in integrative genome viewer tool. Okay, so there we are uploading our sample, we are giving that which organism we are trying to like see the data. Okay, and then we will mention that which chromosome. So we have option to select the chromosome number, and then we need to give the position of the chromosome. Like here you can see chromosome 12, and then the position is 92356 nucleotides. After this number of nucleotides, we have that gene specifically. So here we like here it is marked with the red color. Okay. Yeah, so we have marked it here, and then we can see that on this particular chromosome, this position, we have this gene. So this is the BAM file. Okay, and this is also the bed file. So that we can see here, we can see the gene is here, and how the samples. So you can see row 1 is here, where we can see different reads in red color and blue color. So this is for one sample, and then next six three rows are for the second sample. Okay, but there are different type of files uploaded here, so that also I will explain you later when we will do some analysis on that how we visualize our data actually. Like this, like one sample is on the top where some reads are there, like for example we say it is reference reads, and then the second sample we see here that it is your disease samples, for example. So you can see in the reference genome, like a reference read on the top, there are reads, right? There is in blue read here at this position, and then two red reads are shown here, then again some reads are there. But when we see right below in the disease sample, there are no reads, right? It means there is some of the nucleotides which are missing in your data on this gene. Okay, so this may be possible cause of the disease, or may play some role because these nucleotides are missing. While in healthy person, these nucleotides or the sequence is there available in your gene VLR. The gene name is given at the bottom. So this way we see that how the data is different. Okay, so this is importance of alignment. So okay, this is the SAM. Yeah, this is PDF actually. Yeah, so yeah, now, okay, so now we have the GTF file format. This is again one file which is used for annotation of our reads. So annotation, okay. Yeah, so we have GTF file, which is with extension GTF, and then we have the sequence ID where chromosome number will be given, source is given for which like organism the data is belongs to, type is the data or the nucleotide that is showing is from section, or is it from coding sequence region. So, and what is the start position on the chromosome one, what is the start position, what is the end position. So these columns will be given in the GTF file format. Okay, so this is used to see that what is the like sequence we have in our data and what position it is given. Okay, based on that we can see what is the alteration in the nucleotide that is happening in our data. Next is GFF file format, which is similar to GTF file, but it is just that some of the columns are like, for example, here you can see the last third column, which is the strand column, which is missing here in the GTF file. Okay, so first I show you GTF file. So here in the strand information is not given, that whether this gene or like nucleotide is on positive strand or the negative strand. It means the sequence is read from 5 prime to 3 prime or 3 prime to 5 prime. So based on that, like some informations are available in one file format, some information available in other file. So based on the requirement or the objective of study, we try to use different type of files, or even sometimes we try to convert one file to other file so that we can get required information for our data. Next is cell format, which where the file extension will be .cel, and most of the time this is for microarray data. So microarray data you might all be this. Okay, so microarray data is like coming from after so oligonucleotide. So what is the difference between microarray data and RNA-seq data is because in microarray data we have the set of array on the lab slide where the all the known DNA sequences, cDNA sequences on the slide, and we will take our sample to this slide, so that in our sample whatever data is, whatever genes are identified or present, then it will like pair with the sample which is already available on the on the cell, and then it will show the different signal intensities or in terms of different colors. So let me show you. So here what is the significance of microarray data is that here we can identify, or we can read or and understand or process the known genes only. Okay, so here we are trying to analyze only the known genes. We will not be able to identify unknown genes which are not yet been studied or not been processed in the sequencers. Okay, so and this type of data use cell format. uh, so this is how the cell file looks actually. So the again the header will be there, version is there, that on which like instrument it is being processed, then or hybridized. So we use hybridized term in microarray data. Then we have the header that will tell number of columns and number of rows. Okay, again after the number of headers and rows are given, and then after that, after header completes, then we have the data actually. So here the intensities will be given. So in microarray data we have this expression of the genes in terms of probes. Okay, so if you are already aware about probes and like gene expression and the probes, then it you are already understanding it. If not, then I will explain it to you further. So here is the microarray data where there will be the microarray chip. So wherever you will see the green color, it means these genes are significantly expressed in your sample, because only the reads that are available in your samples, those are only showing the bright colors. Okay, so based on these signal intensity, so these are the color signal intensity is higher, it means that gene is identified in your data, and based on these signal intensity we try to like identify and calculate the expression of the gene. So in one gene there can be multiple probes. Okay, so and then for based on the sample types, we will have this type of tabular data in microarray data when we are dealing with the cells files. Okay. So now databases. So you might already know there are biological databases. So one of and the most famous is NCBI, where we have different types of data like RNA-seq data, single cell RNA data, DNA data. Okay. So if you have not already used, then you will use it further. But there are, let me list you few types of data. This I will move faster because we have less time, and like this is just theory. So database and everything that we need to explore, that is part of a practical session, so that we will explore in practically also. So I will see if I have time sometime, then I will try to explain you. So within NCBI database, we have the microarray data also. We have SRA database. So within my NCBI, we have multiple type of data, independent database also, databases composed or compiled within NCBI. So there is Geo, SRA, GenBank, PubMed, and many other databases which are compiled within NCBI. So we can, like PubMed, from PubMed we extract published papers and all. Then GenBank is there from where we can try to like retrieve DNA sequences. SRA is for DNA and RNA sequences. Geo database is for microarray data and also RNA-seq processed data. So this is how the interface looks. When you go on the all databases, you will see list of all the databases which are compiled within NCBI. So this is geo expression database, where we will get the microarray and RNA microarray raw data, and also we get RNA-seq process data, exome-seq process data. So that also we can get from this geo database. Okay. So I will show you practically. Let me just show you. I will just move it faster. This is ensemble database, where also we can, there are different, for different organisms we can get data. Like here at the bottom you can see there is a human organism given, then there is mouse, there is zebra organism, which is like, I have not shown it here, but you will explore. So maybe I will, when I will share you slide today, you can go through these databases at least once. Just go to the interface and try to explore that what type of data are there stored, because it is important. You need to know that which database has what information, so that whenever you have to do some analysis, you should know that from where you are going to get the data. And I will ask before our practical session from you that like from where we are going to get that data which we are going to process. So let me see like if you are following with the lecture or not. Okay, so this is ensemble database, and then the basics is that there are tools, different type of tools. So and within this database there are raw data also in ensemble. Okay, and some tools are there like Blast, Biomart, and VEP. So this VEP tool, I am just marking it here. So this VEP tool is one of the tool that is used to analyze our VCF file. The VCF file, you remember, right? We have just discussed that VCF file is there for to identify different mutations. Like so, this VEP tool will help you in ensemble database. VEP tool will help you to analyze the mutation data if you have the VCF file. Okay, so just go through these databases after the session. It will be very helpful for you to like see the information whatever is stored. Then there is array express data, where you will this is the interface where you will search for the whatever data you want to search. You can just type that, like there is no strict keyword for searching the data. Okay, just mention it like diabetes or cancer or anything, whatever you want to search, like lung cancer, you can just type. So whatever information is there, these databases are very like interactive and flexible with the keywords. So try to explore these databases. I will like I should recommend, because this is they are very interesting, and information given there is a training section also within this array express. Okay, so and also it is in NCBI, so you can also go through their training like programs. They have some good programs, maybe some are open source and some are like paid. So these are also good, but now we are discussing with data, so you can just get data from here there also. Okay. Okay, I'm just forgetting this is PDF. Yeah, now UniProt. So UniProt is for protein-based sequence, and if you want like protein-based sequence or the functional information, or if you want to annotate your protein-based data, then you can get information from UniProt. Okay, so since we are like this program is about genomics, but you can also explore this data, this database. It is also very interesting because genes are all ultimately like driving the proteins, right? So you should know that where your gene data is leading. Then again, there is a PDB database from where you can get like 3D structures of your like protein molecule, or your like plant. It is derived from different organisms, so this is composed for different organisms, not just the human based data, even for plants also. So you can just go through. Next is PubChem. So this is for the drug or the chemical compounds or the drug compounds. So when we do the gene based and gene sequence analysis, ultimately we try to identify the drug targets, which are because the gene that is causing the disease, we need to identify the drug, right? The right drug which is targeting our gene to fix that issue that it is causing due to gene expression alteration. So this is one of the database where you will get the drug. Oh my God. Okay. UniProt, then PDB, PubChem, then this is the plant-based data. So plant sequences, you can get it from USDA, and this is the interface. So just explore it. And then UNCON database is also there for to get the genome for the woody landscape plants. So there are different like categories in the plants also. So this is specific for woody landscape plants. So if anyone is interested in plant genomics, so go through this database, explore what is there for you. Then plantless database is there, just explore it. Brenda is there for the enzymes repositories, so you can just get there in the like different ligand structure, metabolic pathways, enzymes structure. So if you are interested is in enzyme based informatics, then you can just explore that. Then some of the cancer specific databases you can see if you are interested in cancer studies, where Cosmic is there, which is the catalog of somatic mutations in cancer. So mutation-based studies, you can explore the Cosmic database. Again, International Cancer Genome Consortium is there. Here you can get cancer-based projects. Here you can see 86 cancer projects are stored here. Then cancer primary sites 1222 are given there. And then you can download the data. All the data sets which is here are open source. And the data which is, I have all the databases that I have shown, everything is open source. Most of the thing are open source. So you can just explore and directly see what type of data are available in these databases. Then there is NCG. There
Hello,
also we
Dr.
have
Prasad.
the reference sequence.
Sorry
Yes.
to interrupt. As per early request, it's a gentle reminder. We are around 7 p.m. Indian time. You have told me to
Yeah,
remind
yeah.
you.
Okay, yeah.
Yeah, please go.
No, no, I am okay. I'm following the clock. Okay, let me just
Okay.
wrap up in 2 to 3 minutes.
Yeah, sure. Please go.
Okay. Yeah, so then we have the NCG. So there is the 6.0 version of NCG. So that also you just go through. Like I wanted to show you all these things like on web, but like due to time constraint, it is not possible. So I'm just suggesting you all please go through, because because there is time constraint only. Otherwise, if you want some of the database, I will try to cover like showing you on web in the next sessions surely, because I want to show you few important things to you. and which is very interesting for the gene expression data also. So these are some like pancreatic cancer database, cervical cancer. So for each data like type of cancer also, and if not even cancer, like even diabetes, disease specific databases are also there. So you can see like for pancreatic cancer, there are few databases which are specific to pancreatic cancer, like whatever data that you want to retrieve, you can get it from there. So you just don't need to like Google what are the data set, or maybe you just not need to go to NCBI and type pancreatic cancer. You are not getting like sufficient number of or your required data set. So you can directly go to a specific database. There you will definitely get some important data that you are looking for. So cervical cancer, and then okay, so the express we discuss. Okay, is my PPT already done? Okay, just a minute. Yeah, so the list of, sorry for this inconvenience. Actually, my PPT was not working fine. So that's why I'm using PDF today, and it is not usual. That's why I'm like confusing and changing the slides here. So yeah, major web resources for cancer genomics are few list are given here, which we use very frequently, where Cosmic is already included here, and other databases that I have discussed are there. You just go and explore things. So here is one database which is C Bioportal. Okay, so this is a very interesting database where it is also a database. You will get the data like process data. You will also, it also works as a tool. Okay, so there you can also process your data even without coding or any other tool. You don't require, you just require C Bioportal, get their data, select their data and process there itself and see some results, so that you will feel confident that okay, you can also do some like genomics work. Okay, so just go through these databases at least once, go through their interface, explore. It will help you. Okay, now we are open for discussion. If there is any question, briefly type it. We have like just 5 minutes. Bhavani, do we have 5 minutes? Hello.
Yes, we can. Yeah, yes, we can have
Yeah,
some
yeah.
discussions.
Please,
It
everyone,
seems
if
the
there
session
is.
is very monotonous. Yeah. Yes, please
Yeah.
go.
Okay. So if anyone have any query, please drop down it in chat box. And if there is any query after the session, after going through slides, so also drop down that in the discussion forum actually, because there we will not have the chat box. Yeah, please, anyone, because today was very like easy session. It is very clear everything. So once you explore the database, it will be more interesting for you, because theories are not usually more interesting. So when you will do practical, I'm sure you will like love the databases and what information they have.
Yes.
Okay, so yeah,
So
okay. First,
I have a
hello.
question, just a moment.
Yeah, please.
It
Yes.
was a nice talk. I was just
Yeah,
wondering,
thank
could you please kindly tell
you.
everybody in a layman way, like what are we going to do basically? So you gave us
Okay.
the information about the databases where we can actually see or download all the databases, right?
Mm-hmm. Mm-hmm.
And what are we going to do in the next few days?
Okay, so maybe you already have got the schedule for the program, right? So there it is mentioned that we are going to like analyze gene expression data, and that too for specific to microarray. So we will be using cell format data. Okay, I've explained different type of formats, but initially we will start with the cell format that we will retrieve from NCBI database, and then those data we are going to process with traditional bioinformatics analysis approach. The pipeline we will be following with different tools that is going to be used. And then we will use another step is that we will be using machine learning algorithms to design a model, so that that model will differentiate between our samples, like two categories of samples if we are going to take healthy and disease samples. So it will differentiate and give us a list of or a set of few genes that are important to us as an important features, and then the common genes that we are going to separate from both bioinformatics analysis, like traditional approach, and also the important genes that is identified by our machine learning algorithm. And then those genes we are going to further explore for functional annotation. We will see what are the biological processes they are involved in, where in the cell actually, in which cellular component they are lying, or where they are active for that specific like process. And then we will further analyze which pathways they are involved in, and furthermore we will explore the survival analysis for those specific genes to see that how significant they are for the disease that we are going to study. Okay, so if there is any specific requirement from anyone, or like any yeah, if anyone wants to or wish to tell that what type of disease they are interested to analyze for this session, then they can just mention it in the discussion forum, so that we will take that type of data only. So we will proceed with that. Okay,
Just
so
one more,
that is clear.
small, just a moment. So another one follow-up question. So, are we going to use
Yes.
R or are we going to use
Yes,
Python?
we will be using R, and like for machine learning, we will be using R as well as Python.
So the next question will be like, so we will be doing this in our personal computer.
Yes, your personal computer. I will be showing on mine, so you can use your laptop or the PC if you have, and then you can process there.
So if I'm not wrong, so what sort of specification we are looking here, because it will be like a big calculation.
Specification for like the machine you are going to use?
Yeah, the machines which we are going to use. So any sort of a specification we are looking up for. Because if I'm not wrong, if we are going to use machine learning, probably we should be using GPU, I suppose. Even though if you want to put pressure on your CPU, probably, I don't know how long the calculation might go for. So that's
Mm-hmm.
why I was
Okay.
wondering.
Okay, so just to simplify the analysis so that I can show you, you can just visually show the how the programming is running, how the results it is giving. We will try to reduce the data size, and then we will process within our interface normal 8 GB machine that we have. We will do that. I
That's
have
why I
12
was
GB, but those who have 8 GB, that will also work fine.
That's why I was asking, because it will be kind of if you could already choose the data sets which
Mhm.
doesn't require a large amount of the resources.
Yeah, no, actually, whenever we are using genomics data, at least 22,000 genes we are already having. That is fixed, right? So if we want to simplify, like give less stress on the machine, and also algorithm works fast, we will try to reduce the data size. So we will see like how when we are processing, how we can do that, and we will reduce the data size so that it is just easy for me and for you to understand. I will try to show you on my machine in the real time that how we are processing that data. So we are not needing as of now GPU. If it is required for any further projects, like we can do that on Google Colab as well for Python.
I was going to say that, but okay, cool. Yeah,
Yeah,
all right.
Uh,
yeah.
Hi,
So
hi,
if
ma'am
there is,
Gazala. I have a
yeah,
question.
yeah, please tell me.
Yes. Actually, I have only one question. How we know that when we are doing the analysis that actually this problem is coming from the interruption inside the FASTA-Q file, or it's the VCF file, or it's the SAM file? I mean, when we
Okay.
are doing analysis, So how we will know,
Mhm.
is there any specific error code we will detect, and we will check that this problem is coming from the FASTA file. So we should look around that.
Okay, yeah. I as whenever we do programming, that's why I was mentioning that programming, if you know, that is like an additional bonus point for you, because when we do with in Linux, right? When we do write the program, so there are some tools which give very proper instruction that this is the error, the flow cell in number is missing in your data, in your FASTQ file, like that. So when it comes, or we understand it by practice, like the more data we are processing, like I have been processing data from 5 years, so I know that if it is telling about flow cell, it is definitely in my header, not the plus, not the plus symbol which is on the third row, right? So it will, you will also learn, like when you practice, you will know that these keywords, that if there is a flow cell number information, if there is, sometimes the tool will tell that at the rate is missing or something. If even if the tool is not telling these things, it is just throwing that error, not runs, unsuccessful run, then also you have to go to your data and make sure that your data is totally fine. Okay, so there are programs to check also that your FASTQ is totally fine, its structure is fine. Then also, then accordingly, if you are totally sure that your file is fine, then only you will skip to the next tool. Okay.
Okay, ma'am. Thank you.
Yes.
I have a follow-up questions that if you have some examples of error from, for example, from the FASTA file or from the GTF
Yeah.
or from the BAN, could you please
Mhm.
make a Word file where you can put some screenshots, so it will be easy for the beginners to learn, and then we can detect during the analysis.
Yes, definitely. I have actually. So I will structure it in a document, and probably I will share you, because in this program, we are going to analyze the macro array data. So maybe the error, or if anything is coming up, you will see it there itself. Okay. But if we are using RNA-seq data or exome-seq data, so that maybe I will structure it in a document and probably try to share you. So because
Okay,
everyone will
ma'am.
not understand actually these things, like the beginners will not be able to understand even like what is this saying. But those who are interested, they can just drop down that they want to see the error. So I can share them the documents.
Sure, ma'am. Okay. Thank you.
Yeah,
Thank you.
welcome. So yes, okay. So someone is Lavanya is asking, can you please suggest any colorectal cancer database? Okay, so maybe did I showed you? No, so I have not listed it here. There are databases for colorectal. I have not specifically with that. So maybe I will try to find few, and I will tag you there in the discussion forum. You will go through. Okay. Hope that will work. And yeah, so rest, I think we are already exceeding with the time. So please drop down your queries. Anything, even if you are going through Ensemble or NCBI, any query if you have, just drop down in discussion forum, because from yesterday also I have not received any query. And I was just wondering if you are able to, you are all following the session or not. Okay. So please don't hesitate to drop down. There is no issue. I am already there. So I will answer your queries. Okay. Yeah, hope that was an informational session, and then we will connect tomorrow. Yeah. Have a
Thank
good
you,
day.
Dr. Gasala, for giving us an overview of all the databases. Thank you, all the participants, for joining us today. We request you all to fill out the feedback form. The link has been shared in the chat. Please give all your concerns. We are ready to take up the negative concerns and give better session in future. So I request everyone to fill out the form, and let's all meet tomorrow by the same time. I request all of you to join us on time so there won't be any lag in the sessions. Thank you all once again.